be here. Hello, everybody. Yes, thanks very much for that kind introduction. I'm not going to say anything about ATDBio other than thank you very much for inviting me, and also a bit of a plug, which is a real plug, which is the technology that I'm going to tell you about today, DNA nanorobots. We've got it in R&D phase, and we've now got some production nanorobots as well that we're using in anger with customers. And for the production nanorobots, we only use ATDBio. So we might even have some data to show you why that is, not in this presentation, but another one. So see me afterwards for that. Okay, so when I first joined this company, the first thing I did was I asked my kids whether I should join a company that was working on DNA nanorobots, and they said, absolutely, Dad, yeah, that sounds really cool. So that was my sole reason for doing it, obviously. But then we found out there was actually some good applications of it as well. It's an assay technology, so it's something for detecting nucleic acids, specifically quite short nucleic acids, around about 20 mers and smaller. We had a look at a lot of different applications for this technology. It may have a lot of applications. It may have applications in vaccines and a number of other areas and diagnostics. But what I'm going to talk to you about today is specifically what we found for RNA therapeutics, where there was a real unmet need. So I'm teaching you to suck eggs probably a little bit here, most people in the audience, but clearly RNA therapeutics has been a growing market over the last few years and has really accelerated in pandemic years. But the number of companies going for what could be called RNA therapeutics has increased steadily over a sort of 10-year period. 15, 20 years ago, there wasn't really so much of a market. Also exemplified by this slide, just showing that there's quite a number of big players, and critically, there are now approved, and have been for some time now, RNA therapeutics drugs in some of the categories you can see on the left there. And this has really driven investment into the market, which means we can do more stuff. That's all good. What I'm going to focus on is specifically bioanalysis of RNA therapeutics, because that's where we found an application of our technology. So again, in my intro, you did say I was a molecular biologist, but it was in the last millennium, so please be kind. I've been in business for most of my life, so no difficult scientific questions, please. But what is bioanalysis for those who maybe don't know? Of course, it's tracking a drug, where it goes in the body, half-life, whether it gets to the right tissues. That's the PK or pharmacokinetic side of bioanalysis. You then have the pharmacodynamic side, which is really what happens to the body when you give the drug. That's the therapeutic effect. We're primarily focusing on the PK side of it. And you've seen from some of the talks earlier today, you've got a little bit of a flavor of that. And when I've been at some of the conferences, been happy to be in some of the RNA conferences over the last year, it's been clear that there's a big issue seeing where these molecules go and tracking them, finding out what happens to them when you put them in the body. And hopefully we've got a solution for that. That's a big problem in RNA therapeutics bioanalysis. So this is a bit of an investor slide, so apologies for that, but this is the overall problem that we've got here in the analytical testing market, problems and solutions. So particularly for RNA therapeutics, it tends to be that the technologies that are used are quite complex. They can be variable. Bioanalysts hate variability. It's the bane of their lives. And they have multi-step processing. In contrast, Nanovery has developed a technique which is easy, super ridiculously easy to use, robust, and in particular, in the last 6 months, we've shown that we can apply it direct to patient samples. I've got a bit of data on that. So this is about as simple as you could get for a slide. Sorry, for a slide showing you an assay, right? This really is it, and you don't even need a slide for it. So you can literally say, there's your sample with nucleic acid in it. Here's your magic nanorobots. You add one to the other, and you get a fluorescent signal in proportion to how much nucleic acid you had in your starting sample. It really is as simple as that. And we can get, depending on the concentration, we can actually have signals in 30 or 40 minutes sometimes. So what are the advantages of the technique that we're using here? So we've shown that it can work directly in biological media. It's enzyme-free, so in contrast to things like PCR, there's no enzymes. Isothermal, so you don't need any temperature cycling, anything like that. Cost-effective, simple plate reader protocol. You just need a fluorescent plate reader, nothing else. You don't even need kind of highly qualified staff, actually. And it's optimized for short sequences. So we found that it works particularly well for ASOs and now siRNA as well. Okay, so a little bit on the IP and a little bit on the technology I'm not going to go into too much detail on this today, but this is in essence what our technology does, how it's built. So you have a target sequence that you're looking to detect. We then have 3 modules that we work with. So we have a detector module of nanorobots, clearly detects the specific sequence you're looking for. We have an amplifier that makes sure that you get a decent signal, so it's sensitive. Lots of our IP is around that, actually the amplification bit. And a responder, and the responder basically separates a fluorophore from a quencher at the end of the reaction, and you get a signal. One of the questions I had when I joined the company was, there's a sort of a 20-year-old paper describing this, and this is the basic technique, and the basic technique is not quite as sexy a title as DNA nanorobots. It's toe-hole mediated strand displacement. Now you can see why we call them DNA nanorobots. So this is the thing that's described in the literature 20 years ago. My question coming to Nanovery was, why isn't everybody using it? Okay. The reason nobody's using it is because that academic technique's not very sensitive. You need to do lots of purification. It's fussy. It's not really usable in anger. So that is the stuff that Nanovery has solved over the last 4 or 5 years. This is the basic principle. This is the technique that's based on toe-hole mediated strand displacement. This is very different to something like PCR. As you can see from the diagram, you basically have an input sequence which has a recognition toe hold, and then a branch migration, and then a dissociation toe hold. So you get an initial binding, and then effectively you get an equilibrium where you're getting a branch migration, and then you displace a strand. That's putting it very, very simply. All of the IP and the work that's gone into this, if you look up toe-hole mediated strand displacement, is getting the right structures to make sure this can be used as a sensitive assay. Okay? That's summarizing a lot of work. So why is that of any use? This slide was pretty much provided. So we've actually got a couple of customers already. So we've got a US biotech customer. I'm going to tell you a little bit about the results that I'm able to talk about. And then we've got a top 10 pharma customer as well. The top 10 pharma customer gave us this slide. So I was like, oh, thank you very much. So they actually taught us about what are the problems in this particular market. And so you can see some of the competing technologies there and the pros and cons. Our assay is already round about as sensitive as the best assay, so we've got the sensitivity cracked. But in terms of the development of the others and the complexity of the other assays, then we're winning hands down. So it's much, much easier to develop our assay. Simple protocol, direct to sample. One of the other things there, you can see mass spec, or LCMS, is there on the left. I'd had LCMS in some of my previous companies, and my first assumption going into this venture was, well, you'll just do LCMS for bioanalysis, that's fine, until I learned that the key thing is the sensitivity is not that good. So that's why people tend to look at all these other technologies. So we've got a big advantage over these others. That's all well and good. Does it actually work? I probably wouldn't be telling you if it didn't work, would I say? So, no, it doesn't work. No. So I've got some platform performance to show you that stuff and show you sensitivity, etc. So first of all, the sensitivity, the data is incredibly tight, as you can see on these assays. And routinely, we're getting low picomolar detection. And that's kind of our average assays now are sort of 0 to 10 picomolar sensitivity. That's round about four or five orders of magnitude better than the original literature where we started. So that's where all the development has been over the last time. You can do it in high throughput, so really small volumes. As you can see on here, 10 microliter sample volumes. This is really useful. We haven't actually honestly deployed it in high throughput, but you could do that if necessary. One of the key breakthroughs that we did was applying the assay directly to biological media. That's a huge thing. So unlike PCR and some other techniques where you have to do an extraction, we can do this directly in media. And you can see here the data showing water versus serum versus plasma. And basically there's no difference in terms of the calibration curves. We've got some more data later on showing you that we've gone to tissue samples. That's work in progress. But so far, it works amazingly direct in tissue samples, which is pretty much what every customer asked us for. This is what this shows here. So this was work we did with our very first customer, which is a US biotech, and they were using homogenized brain tissue. We have all these other tissues coming, heart, kidney, liver, cerebrospinal fluid, and other things that people have asked us to do. but they were specifically interested in brain tissue. Their application was in brain, CNS application. And this was amazing results. We were basically doing like this. We were thinking it was going to work. And only about 3 or 4 weeks ago, we got that result. It works absolutely beautifully. You can see the calibration curves. So brain tissue, no problem. You can detect it directly in brain tissue. Specifically for RNA therapeutics, so you've heard earlier in the day about how these RNA therapeutics have various chemical modifications to the backbone. You can see some of them here. That can be a particular problem for assays. That means that some assays literally won't work on them. PCR, you have to have a workaround. You can get it to work. You have to do something called splint ligation. These guys know about that quite well. So you can get it to work, but it's hard. Other techniques are then variable between different backbone modifications. Our technology doesn't care because the nature of the assay is based on binding to the target nucleic acid. So backbone modifications make very little difference. You have to have a different calibration curve, but it works fantastically. So we then also, for those interested in siRNA, we did a modification and got that to work the last couple of months. So primarily the assay was working on single-stranded targets and was a problem for double-stranded because you didn't have any melting. We've solved that as well. So we've got a new protocol whereby we do one extra step with siRNA with double-stranded targets, and we can get it to work for that as well. You can see the calibration curves on the right. So that's a big breakthrough as well. Specificity. So we've kind of cracked this quite early on. This is a study on LET7. So this is microRNA, so not a therapeutic, actually a biomarker. But microRNA 122, this one, LET7, has a number of isoforms that you can see there on the left, A, B, C, etc. And these isoforms have various different nucleotide changes. And but you can see that right down to only having one nucleotide difference, that's LET7E, you can get about 10-fold selectivity over that. We have then been asked, it's not in the presentation, but we've then been asked by various companies, can we do metabolites? And of course, with RNA therapeutics, metabolites tend to be N-1, N-2, to which our head of R&D told me, no chance, about a year ago. And I wouldn't say we've cracked it, but we've now got a nanorobot that does an 8-fold differentiation with an N-1. So metabolites may be coming soon as well. And that's going to be very big. So the key thing for any bioanalytical test is actually reliability. And this was something we came up against with our first large pharma customer, and they went straight into, right, we're going to test this against ICH M10 guidelines, which is the gold standard for bioanalysis for reliability. We did that, we went through it all, and I mean, it just beats absolutely everything, no problem. As you would imagine, because it's such a simple assay. So the validation of this is incredibly easy. This consistency is amazing. So past that with flying colors, no problem. Multiplexing, so the assay also works quite well if you multiplex with different dyes. This isn't a particular advantage over other technologies, but this just shows that there's no kind of system crosstalk between different nanorobots. We can put different nanorobots in together and look for different targets and detect those targets separately from each other. Okay, so that's kind of platform performance. Just moving on to case studies now. And these are some things that we've done in anger with customers. So this is reiterating the point about conjugates. So first of all, you've got an ASO with one set of chemical modifications on the left there, ASO1. Got ASO2 with different chemical modifications, still detects that the same. And then interestingly, the third here, this particular company was using a small molecule conjugate as well to add, and having a look at what happened when we did that, still the same. Again, because the technology is based on binding to the sequence, so the actual assay detection is aligned with the thing that you want the therapeutic to do, which is bind to a target, right? I can only show you a little bit of this, but this was like one of those days you get when you work for these companies, when you get an amazing result, and it's like a lifesaver result for the customer that you work for, and a big breakthrough, and you didn't know whether it was going to happen or not. This was champagne day. So we're working with this US biotech, as I mentioned. they're trying to get their ASO across the blood-brain barrier. They'd used every single other technique they could possibly think of. They couldn't even detect the normal ASO, let alone show that their carrier technology, so they're complexing it with another molecule, could improve the delivery across the blood-brain barrier. So our technology did that. It showed that it went in, and it showed that their technology enhanced the delivery, which unfortunately, you'll have to take my word for it, because I can't show you any of that yet. But that was, yeah, jumping up and down and a very, very happy day. So that's the entire raison d'etre of their approach, is to show that they can improve delivery of this drug for a particular CNS application. So they like us quite a lot, which is nice. There's a third application here. So I mentioned at the start that the company had looked at other applications, and there's a number of other different applications, some of which are shown up here. One big area, of course, in addition to bioanalysis, is diagnostics. For diagnostics, you'll need, diagnostics is challenging, which is one of the reasons that we've pivoted slightly away from it, and requires quite higher sensitivity than we've got right now. But we do kind of have the first data from what looks like a diagnostic application. So this one was microRNA 122, and this is in drug-induced liver injury. So microRNA 122 gets induced with paracetamol-induced liver injury. And you can see this one is really PCR. You can use PCR to detect it, but we're showing that our assay is basically equivalent with PCR, and you can detect it just as well with our technology. So that's in liver toxicity application. Ongoing innovation. So what are we doing in the future? Well, we're always driving sensitivity is one thing. This is one of the things that large pharma customer number one asked us for and described it as the holy grail if we could do it. So I mentioned PK and PD as the two halves of bioanalysis. And we've got it on a later slide, but their head of BioA basically said if you could do that in the same tube, that would be the holy grail. So we do PK and PD in the same tube. In other words, you're looking at what's happening with your drug, whether your drug's got there, whether it's still there, and you can look at a biomarker that says whether it's had an effect in one sample. Okay, so we're not quite there yet, but here's a kind of an example of where we're doing that, where we've got a detection of an ASO, a chemically modified ASO, and we're also looking at microRNA 122 with a different fluorophore in the same sample. So that's a marker of toxicity. In order to do that more generally, we would have to, of course, do it for mRNA. mRNA is longer, and we'd have to have better sensitivity. Both of those, we believe, from the innovation that we're doing now, will be possible, but it can take us probably a year to solve that. Detection sensitivity, I've mentioned a few times. So we've got routinely low picomolar, that's no problem. The best we've ever got is kind of a few hundred femtomolar. Given the innovation and given the progress the guys have made over the last year, I'm pretty confident they're going to drive this further, and we're going to get even more sensitivity, at which point we could think about cracking PD, which would be fantastic. Okay, so in summary, works directly in biological samples, fast, very simple workflow. It's built for small sequences, ASOs, siRNA, microRNA, and it suits a wide variety of chemical modifications. We've got this sort of slightly cheesy strapline, Americans love this, on the right. Let's say high school student, I can't do it in an American accent, not credibly anyway. High school students can successfully conduct this experiment in their first try. So it's not a cheesy strapline because we've done it. So we've had secondary school students come into our labs, we've taught them how to do it, and we've had them doing it. That's how easy it is to do. We are based in Newcastle, so come and say hi if you happen to be up there. I'm not, I'm just 20 minutes down the road in Oxfordshire. So I'm popping up there quite a lot, as you might imagine. Amazing place, so it's a biosphere. I actually did my PhD in Newcastle, where that site is used to be a brewery. So it's completely amazing. It's like a mini Californian biotech now, which is, I was amazed when I went there. So that's pretty cool. Yeah, so you've seen and heard a little bit from me. Absolutely amazing team. I mean, this is why I do these things, because you've got an amazing team of young entrepreneurial scientists who've spun out a company and have developed this out of a university. and are the experts in it, and pretty convinced that no one else in the world is within 5 years of what they've done. So that's handy for investment. Just something on the last slide. So again, some sound bites. I've talked about a few of these already, but we've had a number of really nice comments from customers and collaborators. One of the some of those have already gone through, so I won't reiterate them. One of the key things that kept coming through was that people were having assays that were taking ages to develop and then took 2 days to run, and then were pretty variable. So it was like, we can beat all of those. So that's pretty nice. And as I say, this first biotech, they've basically got a fundraise based on what we've shown them in terms of the delivery across this blood-brain barrier. So, and then hopefully going to have a therapeutic for a pretty nasty CNS application as well. So we get the kind of life-saving thing in there as well at the end. And that's it. Thank you very much.